You've discussed the mechanistic ability of red-eyed tree frogs to respond to vibrations. Has there been any study of the brain in these embryos and its physiological changes?
So we really do not know what is going on in their tiny brains. That is like a very interesting question, and at some point we may get to that, but we're not there yet. Yeah. So I think, I mean, clearly there must be something going on in their brains, right? Because this escape hatching response is not just like a simple reflex, right? Like, so, animals have reflexes, right? So like if someone, hits your knee, your leg jerks, that kind of thing. We use a reflex when we're studying this. So there's a reflex whereby your muscles around your eyes will move your eyes in relation to actually vestibular system input. So like when your ears are telling, just by a very simple, set of neural connections, oh, your head is moving.
So this is a very simple reflex, causes eye movements that compensate for head movements to stabilize the visual field, depends on vestibular function. So we can put a newly hatched tadpole in our custom-made tadpole rotator, turn it, take pictures of its little face, and then measure the eye angle, thank you many undergrads, and calculate the reflex strength from that curve fit. So this shows VOR amplitude across age for 5 different zip shifts, and we see that it increases dramatically at 4 days, matching the escape hatching ontogeny.
So imagine I'm a tadpole, right? Then my eyes are going to be on the sides of my head. And if I am being rotated, okay, so then now like my visual field is like, whoa, changing, right? So if I rotate the eyes like opposite to the way my head is moving, that kind of stabilizes the visual field. That's the vestibulo-ocular reflex. And it's not like they're thinking about it. It's just, it just happens. They just do it. It's a reflex. So hatching in response to these cues, that's not a reflex. That is something that requires more information processing, more integration of information from different sources, potentially more evaluation of risk, essentially, based on the information that's coming in, possibly information sampling, like how long do I wait, kind of to understand what's going on before I make a decision, all of these things. So it is something that requires some brain involvement.
I am trying to understand what are the underlying mechanisms behind the hatching plasticity, because the hatching plasticity happens in a lot of species, in very different species, but in that case, in Agalignis calidrius, the process between when they receive the cue and when the output is generated is very quick. So that means that the process or it's making it in a brain. It looks like it's in the brain because it could be by hormones, but the hormones acts more slower than the innervation. So I'm thinking that when they receive the cue, they very quick process in the brain and make a decision. And then they create an output. And the output is release the enzyme and the shaking behavior to get out. So I am trying to see if there is innervation around the hatching glands. And the first step to try to see if there's something there is use an antibody that marks the synaptic vesicles. So that could mean that there are synapses in these glands. It's a little bit hard to work with a very big tissue because I am trying to look in a very small gland or cell, so it's very hard to see now. But apparently there is something there behind the hatching gland. So I am not sure if the synaptic vesicles are in the hatching cell or are just around, maybe in another tissues or something.
So, like the fact that they're putting together these different pieces of information, yeah, like to me that says that, you know, this is something that has to be involving like a bunch of different neurons communicating with each other in the brain. It's not just like bing, 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 you know, simple reflex arc.